so we'll take a look at number 4. So this is a good answer here. You put these in parentheses to show that after hydrolysis, the sequence would not be clear. But we would know there were 3 separate fragments. That's good. Okay. So these are good answers. So if you use trypsin and a fragment doesn't have lysine or arginine, what does that tell you? If you use trypsin and you get a fragment that does not have arginine or lysine, what does that tell you?
That means it's the part of the last. It's the last part of the peptide chain.
From the carboxy terminus. Good. Because otherwise, since it couldn't be produced, as you can see from this example, normally every fragment has a lysine or an arginine because trypsin cuts after lysine and arginine. The only way to have a fragment that doesn't have lysine or arginine is if it's left off, if it's just cut off at the very end. Okay, good. That's a good answer for number 4. That's a good, careful answer for number 4. So let's take a look at number 5. All right, that's good work for number 5. It's important that you realized when you couldn't figure out anymore. So you knew who comes first, but you can't figure out exactly who goes in these last 3 positions. So you just wrote down the possibilities down here. So it's important to know when what you can't figure out as well. So that was a good, careful approach for number 5. Very good. Let's take a look at number 6. Okay, so let's see, that's number 6. Excellent. So here you figured out a complete tripeptide. So excellent. That's good puzzle doing to work out that entire structure here. Very good. That looks like a good, careful approach on this. One thing here that doesn't quite make sense. This here is not really a reasonable way of writing it because this makes it look like this is the sequence. Is this the true sequence?
Which one?
What I have in a box, is this the correct sequence?
No, it's not.
So that's not really a safe way to write this. That could cause confusion. So if you had to write something, you could just write you could write this if you want to really write it out. You have to write it this way because when that was something you wrote at the beginning of the problem. At the beginning of the problem, you had no idea who went in any of the positions. So rather than writing it like this, where we pretend that we know who goes in the positions, a better way to write out that first step, if you wanted to, would be like this, to show that at the beginning you just don't know. All you know at the beginning is that there's 3 positions. That didn't mess you up on this problem, but this was a pretty easy problem, right? As the problems get more difficult, it's important to have better notation. Okay, fair enough. Also, the way you wrote it here is a little bit misleading. You made it seem like you applied trypsin to the amino acids. This makes it seem like trypsin was applied to the amino acids. Again, if you wanted to write it, a better way to write it would be to say that the trypsin is applied to the original peptide.
Could I do trypsin / hydrolysis?
That sounds fine to me. So if you wanted to write that out, this would be a better way. We don't want to pretend that we know what we're starting with here, but we know we were starting with a tripeptide, not with separate amino acids. So this would be a better way to write the information in part B. This is a better way to write the information in part B. Again, that didn't mess you up on this problem, but when the problems get more difficult, it's better to have better notation. In any case, you did work out a good answer here at the end. And notice here, I wrote this the same way as it was written in the sample problems you sent me. Usually, your instructor doesn't really mention the hydrolysis if they're using things like trypsin, but it's implied by the way this is written. Okay, very good. What type of molecule is this?
Tripeptide.
Excellent. That's right. And what type of molecule is a tripeptide?
Protein.
Yeah, well, a very, very small protein. Very good. What type of molecule is this?
A free amino acid.
Excellent. What type of molecule is this?
An enzyme.
Excellent. And what's a larger category that it belongs in? A more general category? Excuse me. Excellent. Okay, let's go on to number 7. That's good work on number 6. But I did have some suggestions about the notation there. So let's look at your answer here for number 7. Okay, so this answer is correct. So that looks like good work. This was a harder question. Here you figured out a tetrapeptide. So that's excellent. I think this is a little bit harder than any of the problems that you and I had time to do in our session. I gave you some, to save time, I gave you some problems in this homework that were harder than we did in the tutoring session. I was hoping that you could figure out how to do them by using the techniques we talked about, and you did here, so that's great. This is harder than the ones we worked on, but you figured out how to apply our techniques, so that's good that you adapted to that. Again, though, this isn't really the best notation. For example, you said here that you started with this sequence, but is this the true sequence you started with?
No.
So it would be better to just write it as dashes, since we don't know what we're starting with here. Also, you made it seem here like first we did the hydrolysis, and then the Dansyl chloride was done on the products of the hydrolysis, and then the trypsin was done on the products of the Dansyl chloride. But that's not correct. Actually, each of these steps starts with the original tetrapeptide. So you don't want to write them as a sequence like this. If you see what I'm saying, you want to write them as separate like this. Notice here I started, each time you're starting with the original tripeptide. So each time you should start with the original tripeptide. You shouldn't make it seem like part B is working on the products from part A. That's important to think clearly about. For example, I'm not going to do it all out, but the step with the Dansyl chloride you could have written like this. We don't know what the tetrapeptide is yet. We're applying Dansyl chloride, and then even though it's not mentioned, it's implied that they're applying hydrolysis, and then the product from that was Dansyl proline. There would be many other products as well. You would get all the other amino acids as well, but those weren't specifically mentioned. The one they particularly mentioned here was getting the Dansyl proline. So this would be a better way to write out notationally part B. This would be a better way to write out part B. Okay. And then for the next part, you might write it like this. We know, at this point you knew that proline came first, and then we used the trypsin, followed by hydrolysis, and from that you got the methionine and this. So you see what I'm saying? We didn't do the trypsin on the products from the Dansyl chloride. The trypsin step started back at the tetrapeptide again. So each step is usually going back to the tetrapeptide. That's a better way to write it. You did get this problem right, but as the problems get more difficult, it's better to have better notation. All right, but like I say, it's good that you figured out how to solve that. That's harder than we had done before. Let's go on to number 8. Okay, well, this one was a hexapeptide. So this is certainly much harder than any of the problems you and I did together in the tutoring session, but you worked the whole thing out. So very good. That's very good. That's going to save us a lot of time. So it's great that you were able to do that as homework. That's a lot harder than we did before, but you worked it out. I hope that at this point you're starting to see that these are like little puzzles, and you did work out the puzzle correctly. Again, this notation is not very good because the notation makes it seem like Part B was done on the products from Part A, and that's not really right. Each of these should be shown as starting with the original hexapeptide. But anyway, this is the correct answer, so that's good. Number 9. Okay, let's turn on your screen. Let's try number 9 again, because I want to talk about the logic for this. So try number 9 from scratch. Let's try number 9 from scratch. Let's work it out step by step, and you can explain to me what your logic and your reasoning is at each step.
Okay. So
Okay, yeah, just go ahead and try to solve the whole problem. Keep going until you've figured out as much as you can. And as you're going, you can explain to me what your logic is at each point.
okay, so this is part B, and we're reacting these well, hydrolyzed hydrolysis, and then Dansyl chloride, and we get a result of Dansyl isoleucine so we know that 2, 3, 4, 6, 7. We know that the first one is isoleucine. And then we react it with trypsin, which is an enzyme. We get this fragment. Okay, so now we know that isoleucine is first. Isoleucine is also paired with lysine, so that would have to be second because it's because trypsin cuts at lysine and arginine.
That's very good. That's an excellent thought process. You know, that's something we didn't specifically talk about, that kind of deduction last time. So that's good that you're seeing that. So what you're doing here is you're trying to figure out where that fragment goes in what you already know. You're trying to figure out which of those fragments matches up with what you already know. You already know that isoleucine goes first, so you're trying to tie that in with the new information. So that's really good that you're thinking about it that way. That's a very good puzzle-solving technique. So far, so good.
And I know that if it cuts at lysine, the one after it, if I have no free amino acids, the one after it has to be something that's not lysine or arginine. And the ones that don't have lysine or arginine, the fragments are at the end, so I can automatically reduce this to the last one. Or I don't know which order it's in, however. I know that this arginine belongs here.
Excellent.
Where it gets cut, and then...
Excellent. That's very good. Again, that's a little bit beyond what we talked about last time, so it's great that you're making that deduction. That's outstanding. Good.
Okay, and that's as much as I guess we can figure out so far. So we're done?
Let's see. Okay, so you made a lot of good deductions here. I also have a couple suggestions or criticisms. One thing that you have to be very careful on these is if we make any mistakes, it can mess us up completely. So we have to be super careful to double check each step. What does that, so I noticed you wrote down TRY.
Yeah.
What does that stand for?
TYR, I'm sorry.
So let's be careful about that. One little mistake like that can definitely cause a lot of confusion. Okay. What is that? What amino acid is that?
That is tyrosine.
All right. That's relatively minor. So look at what you wrote. I'm not quite understanding what you wrote in the last two slots. What did you write underneath the last two slots?
Should I have probably...
All right, that might seem like a small thing, but we need to write down every single thing we know. I didn't quite say that last time, but the key problem-solving technique is to write down every single thing you know. Our key problem-solving technique here is to write down every single thing we know, single thing that we understand. Okay. So now we're ready to continue. So are we done with problem 9, or is there more we can do?
That's it.
Did you not use part C? Or wait, am I confused? Wait. Oh. Crap. I think I thought you messed up, but that was me that was confused. All right, 1 second. Sorry about that. Let's see here. Where was I? All right, I apologize. I was trying to point out where you made a mistake when actually you did fine. It was me that was confused. You got as far as you could on that one. Okay, fair enough. So one second here. Okay, you did that fine. I would again make a suggestion though, like when you're doing when you're trying to write down what's happening in part A. Well, I don't know. It seems like you're understanding this correctly, so maybe that's not too important to worry about. Maybe we won't focus on that too much there. So. Again, I was just going to mention that for part A, can you turn your screen on again?
Oh, turned off? I know.
Thank you. Yes. Yeah. On these problems seems a little bit confused. For example, for part A, the most logical notation would be this. So it seemed like you were writing the amino acids before the hydrolysis, but we don't actually know what the amino acids are until after the hydrolysis. So this would be...
Should I just write hydrolysis and then an arrow going down to the amino acids?
That's fine. Yeah, you can do that. And then when you go on to part B, you don't want to write it like this. because this makes it seem like you're applying the Dansyl chloride to the separate amino acids. If you're going to do that, you should at least like put in a line to show now you're doing something separate with the Dansyl chloride. Again, it might be clearest if you actually wrote in the dashes to show that you're going back to the heptapeptide. And then you write the results from that. And then when you use the trypsin, again, it shouldn't just be written as a continuation of this. It's a separate analysis that you're doing, again, starting with the heptapeptide. Okay, in any case, you actually did that correctly. I was the one who was getting confused. So let's move on to the next problem. Yeah, okay. So this is something we didn't talk about, chymotrypsin, but I was hoping that you could learn it kind of from this homework problem. It looks like you did a good job of that. What type of molecule is chymotrypsin?
An enzyme.
Yeah, another enzyme, another proteolytic enzyme that cuts peptides. And according to this, chymotrypsin cleaves after tyrosine, phenylalanine, and tryptophan. What do those three amino acids have in common?
They're aromatic.
So it's not surprising that they're all treated the same by chymotrypsin. Did you get a chance to get in touch with your instructor and see whether you have to have those cleavage sites memorized?
I think so, yes, we do.
Did you confirm that with the instructor, or did you just...
I haven't gotten back, but I did.
Well, the problem is that the table in your book has 10 different enzymes. I'd be amazed if he expects you to have all 10 of those memorized. So you should definitely contact him to see which ones you're expected to have memorized, unless you think you know. I mean, which ones do you think you have to have memorized?
The ones that were on the sheet. Then
Oh, okay. That's certainly possible. That's certainly possible. So that would be a good guess. It couldn't hurt to try to confirm it with your instructor, though. It's possible they don't expect you to have any of them memorized, because on the sheet they gave you the cleavage sites. So it couldn't hurt. I don't know if you have your instructor's email, but it certainly couldn't hurt to try to figure out exactly which ones you're expected to know. Okay, so in any case, so after chymotrypsin, will we still have will we have intact peptide fragments or just separate amino acids?
Separate amino acid.
After the chymotrypsin, but before the hydrolysis.
Intact peptides.
So do you see these parentheses don't really make sense here?
Correct.
So it would be clearer to leave the parentheses out. You drew the dashes. Remember, does dashes mean a sequence or a composition?
Dashes means a sequence.
And you were right. Here, there's still sequences. So at this step, we should be leaving off the parentheses. You wrote this step correct. This step is correct, but this step shouldn't have the parentheses. Okay. And then you figured out something here. If a fragment doesn't have tryptophan, tyrosine, or phenylalanine, what does that tell you? After you use chymotrypsin.
It belongs at the end of the peptide chain.
You can see that here, because when you use chymotrypsin, every single fragment has phenylalanine, tryptophan, or tyrosine, except the one that's at the very end. Okay. Okay, so actually, that except for this notation, that was very good work at number 10. Very good. Okay, here's the, so now let's turn on your screen and work on number 11. So let's try number 11 again on your screen. So let's start this from scratch. Just try the problem again from scratch and show me how you would work out each step and explain what your logic is. Okay, so I'm going to make a suggestion. You're probably going to be better off trying to figure stuff out as you go. On the right-hand side of your screen, write out the amino acid composition of the entire peptide. On the right-hand side of your screen, maybe at the top right-hand, write out the entire amino acid composition. Not the dashes, but the actual composition of the amino acids on the right side of this right half of the screen, please. Excellent. Now I have a suggestion. Instead of writing lysine sub 2, write the lysine down twice. And let's double check. Do we have the right number there? 1, 2, 3, 4, 5, 6, 7, 8. Okay. Now you can write your dashes below that. Now, from now on, every time we figure out who goes in a position, we should write it in that chart. And excellent, you saw what I was going to say. Every time we know where someone is placed, we can cross it off of that list. Okay, so every time we figure something out, we should be going over there and writing it down as we go. You don't want to go through all the clues before you start figuring stuff out. You want to be figuring stuff out as you're working through the clues. Okay, so let's continue. Okay, I'm going to interrupt you again because right now it looks like you're not quite taking the advice I gave you a second ago. Remember that before we go on to the next clue, we want to figure out as much as we can from the trypsin clue.
Okay.
Okay, that's very good. It's good. One of the most important things you did there is you took your time. It's important to really slow down and take your time because this is puzzle doing. It takes a time to have the insights that we need. So you saw that isoleucine and the tryptophan fragment had to be at the far end because it didn't have any lysines or arginines in it. That was good. And also it looks like you're seeing that all the other fragments have to end in lysine or arginine. So you knew that the lysine pro fragment had to end in lysine because this enzyme cleaves after lysine. So that was a good deduction as well. Okay, so now we are ready to move on. Very good.
Okay.
Okay, excellent. Now, I don't know if you remember, but when you did this before, you didn't figure out the whole peptide, right?
Yeah.
Okay. All right. So when you the answer you gave to this when you did it as homework was not correct because there turned out to be deductions you were missing. But it's great that you figured it out this time. I didn't give you very much help on this. I didn't give you too much help. So that's good that you worked that out. That's excellent. So how do you know that the isoleucine is at the end?
Because it's free after chymotrypsin.
So the key thing is we know any fragment when you use chymotrypsin, any fragment that doesn't have trypsin or tyrosine or phenylalanine must be last, while that isoleucine fragment doesn't have trypsin, phenylalanine, or tyrosine, so it must have been last. That's one way to see it. And then you know all of the other fragments have to end in trypsin, tyrosine, or phenylalanine. So that's how you knew that the first three had to be valine, serine, tryptophan. The serine has to come before the tryptophan because the chymotrypsin is going to cleave after the tryptophan. That's the only way to get that 3 amino acid sequence. Okay, so that was great. Just remember that you weren't quite figuring this out when you were working that before, so you should ask yourself, what did you do differently this time? I'm not quite sure what we did differently. Maybe it was just that I was kind of prompting you to figure out as much as you could after each clue. Don't wait till the end to figure stuff out. Figure out as much as you can after each clue. But otherwise, that was a great use of notation. Again, I would just say where you do you see that point to the arrow where it says you have the down arrow on the left and it says Dansyl chloride?
This one?
Yeah. I just think that's not great notation because it makes it look like you're applying the Dansyl chloride to the amino acids above it. So let's write a big horizontal line above that Dansyl chloride just to show that's a new step starting from scratch. And again, let's do the same thing above the trypsin arrow to show that that's a new treatment starting from scratch, not on the products from the previous step. And let's write a big arrow above the chymotrypsin to show again that's starting from scratch, starting from the octapeptide. We're not doing that on the products from the original step. Otherwise, that notation can be misleading. Okay, very good. All right, can you see my screen?
Yes.
Okay, so here's what you did when you did this on the homework. So there was a couple of deductions that you were missing here. I'm not sure exactly what was going through your mind here, but you didn't quite figure out this order here at the end. Let's see, trypsin, lysine. So it should have been clear that this was a lysine from this fragment. From this fragment, we should have known that this position was a lysine. And then we should have known that this was the proline and the lysine. I think what went wrong again is that you waited until you'd looked at all the clues before you started figuring stuff out. That just is more confusing. So this time we were able to get out more information. Okay, good. Okay, well, it's great that you were able to work through all those problems. That definitely is going to save us a lot of time today. If you hadn't been able to do those as homework, we'd have to do them all together, and that would use up most of the session. So it's great that you went through those. You figured out a lot of techniques there on your own that we hadn't talked about, so that was good work as well. So let me write something down here. Okay, so when you're ready, turn on your screen and try solving this problem. Try to work out step by step and explain your logic as we've done previously. Oh, looks like I left something out here. I left out this for part A. One moment. Okay, so that's what I meant for part A. So when you're ready, you can turn on your screen.
Where are you making these problems up, or are you getting them from somewhere?
I made them up.
Wow. Okay.
Okay, you done?
Yep.
All right. Okay. So very good. So it looks like you worked out the puzzle here again. So after using the chymotrypsin, we knew that proline lysine was last because it didn't have one of the aromatic residues in it. And you also figured out, you also knew that tyrosine and tryptophan have to come last in each of their fragments from chymotrypsin. So that gave you a lot of information. And then the key thing when you use the trypsin is that you try to see how the trypsin results overlap with the chymotrypsin. So you must have seen here, so you knew that proline had to be the last because proline didn't have arginine or lysine in it after the trypsin. And then you just had to ask how those two remaining fragments overlapped with the work that you already had. So for example, clearly, point to the serine, arginine, threonine, tyrosine in D. You knew that that had to come first because it has serine in it. And that pretty much told you, when you used the previous results, where everybody went. And then you knew that the lysine, threonine, tryptophan had to be in the middle. And so when you overlaid that on your previous ideas, you were able to figure out where pretty much everyone went. That was very good. That was an excellent puzzle-solving process. So those problems are not easy for everybody, so it's good that you worked that out. Okay, one moment. Let me just warn you again, when you take the test, be extra careful to double check everything you're writing down. Because if you make one single careless mistake anywhere along the ways, all of your work will be wasted. For example, if you ever miswrite what the amino acid is that you're working in, change one amino acid to another, or you make a deduction that's not really true, all the rest of your work will be meaningless. So you didn't make any careless mistakes here, but remember to be extra careful, especially on the test, to double check everything you're writing down as you're going. You don't want to have to wait until the end to find mistakes. You need to catch your mistakes as you're going, because if you make a mistake, you'll end up wasting a lot of time on the problem. That didn't happen here, but that's something to worry about. This might seem like a weird comment, but in kind of a weird way, these problems are kind of similar to the spectroscopy problems from organic chemistry that we spent so much time on. Actually, they are pretty similar because they're both about trying to take experimental data to figure out the structure of a compound. So they're all, again, it's a kind of puzzle doing. This is a kind of puzzle doing like the spectroscopy problems we spent a lot of time on last semester. And again, one thing we emphasized was take your time and try to avoid mistakes, because if you make one mistake, that's going to lead you to a lot of problems. Okay. So all of the peptides that you and I have discussed previously have been linear. However, it's possible for a peptide to be circular. It's possible for a peptide to be circular, forming a circle, which is what we have here. So based on the sample problems your instructor gave you, they want you to be familiar with that as well. So the earlier problems we did were all linear, but this is going to be circular. So we have to specify now what our amino to carboxy direction is. So here I'll just specify that the amino to carboxy direction is clockwise. If you have a linear peptide, we know we put the amino group on the left, the amino terminus on if we have a linear peptide, we know that the amino to carboxy direction is left to right. But terms like left and right don't really apply to a circle. So for a circle, you have to specify. So I'll just keep specifying clockwise as the amino to carboxy direction. Okay. All right, so here we have a circular peptide. What do you think would happen if you reacted this with Dansyl chloride? That's a little puzzle for you. What would happen if we reacted this with Dansyl chloride?
Affected?
Could you repeat that, please?
It wouldn't get affected?
Excellent. How do you know? What's your logic?
Because there's no start to a circle.
What does Dansyl chloride attach to?
The N-terminus?
But there is no N-terminus. So yeah, a circle has no N-terminus, so Dansyl chloride here would give us no reaction. So let me ask you, when you're doing the problems on the test, how can you tell whether it's a circular or a linear peptide? What would be a clue that you could look for to see whether it's a linear or a circular peptide if you're doing problems on the exam?
If there's no N-terminus. Or C-terminus either.
But how would you know that there's no N-terminus? They won't just tell you that directly.
Dansyl chloride would be able to react to it.
So if they tell you that Dansyl chloride gave no reaction, what do you deduce from that?
That there is no N-terminus.
Which probably means what
It's
about?
a circular peptide.
That's right. Okay, so that's the first thing that we learned here. All right, what would we get if we applied chymotrypsin to this?
We would have a separation between trypsin and glycine, and I wouldn't know. Well, would trypsin become free? I guess
This trypsin?
both trypsins,
Why would you say that?
because chymotrypsin cuts in front of. Wait. No, that's tryptophan. Chymotrypsin would cut in front of and behind tryptophan,
Why do you say that?
because I wouldn't know which way is in front of trypsin because it's a circle.
Okay. Well, remember that in front of and behind are just shorthand anyway. When we said that we were cutting behind amino acids, did we mean we were cutting on their amino end or their carboxy end?
We're cutting on their carboxy end.
And if you're cutting before an amino acid, which end are you cutting on?
The amino end.
Just because the convention is that amino comes before carboxy. So it's true that before and after doesn't really have a meaning here, but each of these still has an amino end and a carboxy end. Each of these still has an amino end and a carboxy end. Maybe.
You know, it's a carboxy, okay.
That's why I had to tell you what my amino to carboxy direction was, because that's not obvious. There's not obvious from the picture anymore. If it was linear, you would know the amino end was on the left and the carboxy was on the right. But if it's a circle, you have to specify what your amino to carboxy direction is. That's why I specified here that this was our amino to carboxy direction. So that should tell you who we can kind of count as before and who we can count as after each amino acid.
So it would cut in front of the tryptophan between glycine and tryptophan, and then it would cut in front of the tryptophan between tryptophan and arginine.
So 2 cuts?
And in front of tyrosine and tryptophan.
Good. To me, I would say it would be after the tyrosine, not in front of the tyrosine, since this is the direction we're reading in. I'm not quite sure what you're saying in front, but it looks like you're thinking of the right thing. So what fragments would we get?
You'd have a free tryptophan, and then arginine linked with serine, lysine, and tryptophan. And then glycine with alanine and tryptophan.
Say that again, please. That last part.
Alanine and tryptophan.
So what should I write here?
Or tyrosine, sorry.
Okay. Like this?
Yes.
And then if we did our normal hydrolysis, we wouldn't know the order in each fragment anymore.
So hydrolysis would essentially just ruin it?
Well, no. Well, the point is we would do hydrolysis on each fragment separately. So here, here we're just doing the same thing we've done previously. Remember that normally on the problems that we've seen, they're not telling you the results directly from the chymotrypsin, which would give you sequences. They're telling you the results after doing chymotrypsin and then hydrolysis. But they're doing hydrolysis on each fragment separately. So you would still know that this is one fragment, you just wouldn't know the order of it. So this part here is supposed to be exactly the same as for the linear amino acids. This is exactly the same analysis as for the linear amino acids. Notice after you use chymotrypsin, do you get circular fragments or linear fragments?
Linear.
So it just turns out, it goes back to being linear when you use that peptide. Okay. Notice that now every single fragment ends in tyrosine or tryptophan, or phenylalanine. So we can't use the trick that we used earlier. Remember that for a linear amino acid, we could tell who went at the end because it wouldn't have a tyrosine or a tryptophan or a phenylalanine after chymotrypsin, but that doesn't work for circular peptides because they don't have an end, if that makes any sense. What would happen if we applied trypsin to the peptide?
If we apply trypsin to the
original octopeptide.
The octopeptide, we would have a cut between arginine and serine, and then a cut in front of lysine or between lysine and tryptophan. And that's it.
So what fragments would you get?
You would get serine, lysine linked together, tryptophan, glycine, alanine, tyrosine, tryptophan again, and arginine. That's it.
And then you'd get the normal results if you hydrolyzed. If you hydrolyzed each fragment separately, you wouldn't know the sequence, but you would know that they came from two separate fragments. Okay, so this is how these types of enzymes work on circular peptides. You have this down?
1 second. Okay.
Okay. Let's go back to this example that we were doing before. Do you remember this example we were just talking about? Here was the results if we'd used chymotrypsin. Suppose that after using the chymotrypsin. So what happens if we react this octapeptide with Dansyl chloride?
Nothing happens.
But what happens if we react it with chymotrypsin first, and then we react it with Dansyl chloride?
Then you get a reaction.
What exactly would you get? So remember that you're going to react with chymotrypsin, then you're going to take these 3 fragments and separate them into 3 separate flasks. Then you're going to react each of those with Dansyl chloride. So then what would you get?
You would have one amino acid with Dansyl chloride on it.
Can you be more specific about what we're going to get?
I don't know which exactly will get the Dansyl chloride.
What does Dansyl chloride do?
It determines which one is the N-terminus.
But how does it do that? Well, what does it do chemically?
I don't know.
I just wanted to go over something that we talked about before. It attaches to the N-terminus.
Oh, okay.
Right. It doesn't know it's the N-terminus. It just attaches to the N-terminus.
Right.
So what would happen when the Dansyl chloride reacts with this fragment?
It would attach to the N-terminus of the arginine.
So that would produce and what happens when the Dansyl chloride reacts with this fragment?
It will attach to the glycine.
And what happens when the Dansyl chloride reacts with this?
It'll be Dansyl tryptophan.
Will it attach to its amino side or its carboxy side?
To the amino side.
Because that's what Dansyl chloride does. It attaches to the amino. And then if you do hydrolysis, you'll get one thing that has a composition like this. You'll get something that has a composition like this. You'll get this. So, you can still apply Dansyl chloride after you use chymotrypsin. And what will the Dansyl chloride tell you? It'll tell you what the N-terminus was of that fragment. You can't find the N-terminus of the octapeptide because it doesn't have one, but you can still find the N-terminus of each fragment using Dansyl chloride. Does that make sense so far?
Yes.
So what would happen if we applied Dansyl chloride to the trypsin after using, suppose we use trypsin first and then we apply Dansyl chloride, what would we get?
We would get Dansyl serine first and then Dansyl tryptophan first.
And then after you hydrolyze them, you would get this. Okay. Leave that down.
Yes.
By the way, do you see here why one of the tryptophans is attached to Dansyl and one isn't?
Mm-hmm.
Because one of the tryptophans was at the N-terminus and one was in the interior. So we ended up with two different types of tryptophan here, if that makes sense. Okay, so far so good?
Yes.
So let's go back to writing down this problem. I was writing down this problem. Hopefully you started writing it down, so you can go back to writing it down. Okay, here's the problem. Make sure you copy it carefully. Remember, you have to make sure you're avoiding careless mistakes, and when you're ready, you can turn on your screen and try solving the problem.
Okay, so
Okay, let's go through this step by step a little bit more. What's what's the first clue in this problem?
the first clue is that trypsin. We reacted with trypsin first, so it should be leucine, arginine.
All right, now let's back up. Take a look at the problem again. What's the first clue in the problem? Trypsin was not the first clue. What was the first clue they gave us about the structure?
Chloride gives no reaction.
What can we deduce from that?
It's circular.
Okay, but you haven't written it as circular.
Mm-hmm.
Now,
should I just erase this whole thing?
Well, that's not supposed to show structure anyway. That was just the composition, right? Your dashes are supposed you have linear dashes, so you should rewrite the dashes so they form a circle. Kind of like I did in the previous problem on my screen. Remember, we're going to try to use this work to solve the problem. You don't need to erase your list of amino acids because that's not trying to show the sequence anyway. But our dashes should show the circular structure.
Okay.
Okay, how did we know it's circular?
Because it doesn't react with Dansyl chloride.
The intact peptide doesn't react with Dansyl chloride, which means there's no N-terminus. Now, you have to write down what you're going to assume is your amino to carboxy direction off to the side, like I did with my circle. So you have to choose clockwise or counterclockwise as your amino to carboxy direction. You may as well choose clockwise since that's what I did on my screen previously. Okay, so now let's continue. We want to build everything we can into that picture. We want to build all our deductions into that picture. And when you start putting things in, you can put the first amino acid anywhere. You can put the first amino acid you place, you can put anywhere you please because there is no start or end. Fair enough?
So leucine.
That's excellent. We know that every fragment from trypsin has to end with arginine or lysines. That's an excellent deduction. And that's also excellent, and that's excellent notation. Unfortunately here we can't write under the line because there's no line, but that's an excellent notation to show that we know who goes in those dashes, but we don't know the correct order.
Okay.
Let me interrupt you. Now, it's good that you tried to figure stuff out before you moved on to the trypsin, and to the chymotrypsin. It's good that you tried to figure stuff out. We want to figure out as much as we can. So here's where I think you made one of those careless mistakes I was warning against. You made a careless mistake and you didn't copy all the information that you know. So what do we know? What information did we get from the trypsin? Go ahead and reread what information you were given. What happened when they used the trypsin? What does the problem tell you?
That we use Dansyl chloride also.
That's right. And what were the results from using the Dansyl chloride with the trypsin?
We had Dansyl glycine also. So then this would be here.
Do you see how you lost that? It's because you used to have Dansyl glycine written down, but then at one point you erased what you had written down and you rewrote it, and you made a careless mistake and you wrote the glycine without the Dansyl. That's what I mean when I say that you have to be super careful to avoid even little careless mistakes like that. So what you should have done is up above where you have the products from trypsin, you shouldn't have glycine, you should have Dansyl glycine, if you see what I'm saying. Do you see where you wrote your products from trypsin? What messed you up is you should not write glycine, you should write Dansyl glycine. Every little thing we write down has to be perfect, or we're going to get messed up. If you don't write that down, the problem becomes difficult or maybe impossible. So remember to double check everything. I think you used to have the Dansyl glycine written and then you accidentally erased it. So you just have to be super careful to write down everything. The thing that might have been weird here is you're probably not used to seeing two Dansyls at the same time, right? You might have assumed there could only be one Dansyl, but that was from the earlier linear types of problems. Those were from the problems where we were applying Dansyl chloride to the complete peptide. Here we're applying Dansyl chloride to the products from trypsin. Well, since there's two fragments, there's going to be two amino terminuses, so there could be more places the Dansyl chloride can attach. Okay, so remember to make sure you squeeze out as much information as you can and avoid careless mistakes before you move on to the next step, and now we can go on to the chymotrypsin. It's good that you are erasing notations that you don't need anymore. That's a good technique as well. But although you want to be extra careful not to erase things while you still need them. But if you're sure you don't need something, it's good to erase. How did you know that was the order for the alanine and the tryptophan?
Because chymotrypsin cuts in front of tryptophan, so it separates tryptophan and lysine in front of it. Hello?
Yes. Can you repeat that logic again?
It cuts in front of tryptophan. Chymotrypsin cuts in front of tryptophan. So
does it cut on its amino side or its carboxy side?
It's gonna cut on the amino side.
Okay. Confirm that in your table or in your notes. Or in the homework. Do you have written down anywhere what chymotrypsin does?
Oh wait, no, because alanine is with this group here, so it's the other way around. That's why. Sorry.
But first of all, let's make sure we know what chymotrypsin does. What does chymotrypsin do?
Chymotrypsin cleaves after tyrosine, tryptophan, and phenylalanine
After them or before them?
after them.
But I think a moment ago you were saying before?
Right, yeah, that was a mistake.
Okay, so you got to watch out for that. So remember, you got to double check extra carefully to avoid careless mistakes. And if you really need to have those memorized, no time like the present, you better get cracking and memorize those if you really think your instructor expects you to have those memorized. Because you can see it doesn't matter how good your skills are, if you don't know what the enzymes do, these problems are impossible. So if you really think you need to have those memorized, you'll need to do that. There's a couple ways to figure this out. I think you could also see your previous ordering didn't make sense because the fragments were not consistent with the other clues that you had seen. All right, let's see what we have here. What do you have written down after the phi? After the phenylalanine?
Isoleucine.
What letter? What did you write down there on the dash? What letters?
ILE. Oh.
Okay, good. All right, so this is another type of problem that you might see. Remember that one technique here is to actually draw this in a circle. Another technique is, remember, you can put the first amino acid anywhere you please. Since there's no beginning or end, you could start by just putting the leucine anywhere you pleased, and then you tried to place everyone else in relation to that. It would be good to try this problem again from scratch to make sure you know how to get this right. We had some problems with this. Remember, you always just want to double check yourself to make sure that your answers are making sense. You could double check this now by checking that it would really give you the right results from trypsin and from chymotrypsin. Okay, good. So here I have two peptide strands. Now, what do you think this represents?
Disulfide bond.
And what's this represent?
A disulfide bond.
And here's another one. Do you agree that these are not the only possible disulfide bonds we could have drawn here? There's many different ways we could have connected the cysteines, right?
Yes.
So one of the problems in the homework is different from all the ones we've talked about. One of the problems gives you the sequence and just asks you to figure out where the disulfide bonds are. It's actually a lot different from what we've been talking about. There's a whole different type of problem that gives you the sequence and asks you to figure out where the disulfide bonds are. So that's a whole other story, basically. I don't know if we'll really have time to go into detail about that. But one thing that we could talk about there is, let's say that we digested this with thermolysin. Let's say that we digest this with thermolysin, and then we hydrolyze it, and then we break the disulfide bonds and then we hydrolyze it. So first of all, where would thermolysin cleave? And we haven't talked about that yet, so you might, no, that's not even in the table in your textbook. Okay, but it's on the protein sequencing problems page. So if you look at the protein sequencing problems, look at the very bottom of the page. Do you see where it tells you how thermolysin works?
Yeah.
What does it say?
It cleaves before leucine, isoleucine, phenylalanine, tryptophan, tyrosine, and valine.
So, what's the first position where it would cleave here?
Phenylalanine.
Between, you mean this one?
Yeah, before
Between
before
cysteine and phenylalanine, or between phenylalanine and proline?
cysteine and phenylalanine.
Okay, where else would we cleave?
Between arginine and tryptophan. Right, between, yeah.
Yes?
Yes.
Okay. Where else?
And between valine and arginine.
Mean here?
Arginine, yeah.
Okay.
Yes.
Where else?
Hold on a second. Between cysteine and tyrosine on the bottom one.
Anywhere else?
And between cysteine and phenylalanine.
Okay. All right, so we're about out of time. One thing I wanted to point out is, do you see that this is a peptide that has quaternary structure? Because there's two separate strands held together by disulfide bonds. So this is harder than the earlier problems because there's a quaternary structure here. By the way, so does thermolysin cleave before or after the key amino acids?
Before.
How about trypsin?
After.
How about chymotrypsin?
After.
It would be easy to get those confused, wouldn't it? So you have to be very careful. Some amino acids cleave before, some enzymes cleave before, some cleave after. You have to be super careful not to get those confused with each other. So again, don't get confused. Some enzymes cleave before key amino acids, other enzymes cleave after. It's important to be clear about that. Okay, well, it looks like we won't be able to get any further with that today. But anyway, we talked a little bit about how thermolysin would attack this. This isn't even in the table, but maybe your instructor expects you to have thermolysin memorized since it's in the homework.